So we have looked at Darwin's theory of natural selection last week. And we've looked at the diagram that represents the entire flow of Darwin's theory of natural selection. Have you looked at bacterial development of antibiotics in bacteria population? Yes, so you see that how we use that Darwin's theory of natural selection to actually describe the whole process of bacterial population, right? From a population that have got all the variations until a population where all of them are antibiotic resistant. Okay, so using Darwin's theory of natural selection. So today we move on to look at how there are three things here. How selection, which is the natural selection you have learned, has been able to causes change in allele frequency. So you have seen that natural selection can cause a change in allele frequency where the favorable allele will be selected for, right? So survival and reproduce and pass on to offspring. So therefore the favorable allele frequency has increased. Unfavorable allele frequency has dropped. So selection has actually affected allele frequency in population. And today we're going to look at two more factors. Founder effect and genetic drift both also can lead to change in allele frequency in the population. Okay. Then we're going to do some mathematics, which is Hardy-Weinberg principle. We're going to use this Hardy-Weinberg principle to calculate allele and genotype frequencies, and then see when it can be applied. So when we can apply this to calculate allele and genotype frequencies. Not every population we can do that. So what kind of population we can use this Hardy-Weinberg principle to find out the allele frequency and also the genotype frequency of certain genotype in the population. So this is today's lesson. So this diagram shows you that in a large population, very large population, where individuals can interbreed, allele frequencies may fluctuate but remain fairly stable. So we already seen before, the graph where the population size will increase, decrease, increase, decrease around carrying capacity. Okay, that average number of individuals in the population is called the carrying capacity. So here you can see that the first generation got 56% are black. Then the next generation, 42% black. Next one, 64% black. Next one, 60. So you can see that the percentage of black color actually fluctuates around a certain carrying capacity. So it's more or less allele frequency, in this case here, black, right? We're looking at black color, allele frequency is more or less constant. So this is for a very large population when there are no changes in environmental factor. Okay, the same environmental factors acting on it, then the population size, the allele frequency remain more or less constant. Now, what happens if the population size is very small? Population size, when it's very small, is subjected to a lot of changes, right? Because if you look at this example in the diagram, the moment one or two birds are gone, then you can see that there's a big change in the population size, because if this is gone, either it migrates away to somewhere or it died, then whatever left is only 4 out of 6. And this is a very big change to the population because the size is very small. Any changes to any of the individual will cause a big change in the population. So in a small population, any factors or incidents or random events, these are actually the so-called selection pressure from the environment. Remember, you learn selection pressure is any abiotic, so it's divided into abiotic as well as B biotic factor from the environment which can affect their survival, right? Any change in temperature, suddenly there is a hurricane, okay? Suddenly there is snow, okay? Or no rain, so it's abiotic. Biotic can be a lot of reasons. It could be predator, eagle, for example. It can be disease like virus, pathogen, virus, fungus, bacteria that can cause disease, so they die, right? Biotic, or competition for food, competition for breeding ground, competition for mate. So all these things will be and can be the factors that affect the survival, so the selection pressure. So in small population, the factors, the incidents, the random events, all included in this selection pressure, have a greater impact on the population of organisms. So this will lead to change in the allele frequency easily. So let's recap. What is allele frequency? Allele frequency is a measure of the relative frequency, or meaning the proportion of an allele at a locus in a population. That means what is the total number of the allele that we're interested in divided by total number of all the different type of alleles, there's an E here, of the same gene. Okay, so if this is small a only, so what is allele frequency is small a, that means total number of small a versus big A allele and small a allele in the population. Okay, so there's allele frequency. Expressed in proportion or in percentage. Okay. Now, just now we talked about the disturbing effect. This is actually the factors, right? The incidents, the events. So that's called the disturbing factors from the environment. So what is the disturbing factors from environment that can change allele frequency of a population? Selection, which is the natural selection and artificial selection. We have not learned artificial yet. We have learned natural selection. Okay. Genetic drift, founder effect, as well as many other factors which are not in a syllabus that are very obvious. We know it will change the allele frequency because mutation means arising of a new allele, isn't it? Which is not there. Okay, mutation. It can be big A, small a, and suddenly something else like what? E. The gene got allele big A, small a, and E because of mutation. Okay. Emigration means this is a whole population, right? This poor population only got A, and then got small a, and then got an E because of mutation. Emigration is some other organism from outside this population migrate and come in and bring in another allele, for example, I, for the same gene. So change in allele frequency, isn't it? Okay. Emigration and immigration. Emigration means go out. So in this population, suddenly this individual with small e goes out. So there's no more small e, right? So allele frequency again change. Okay. Gene flow. Gene flow means interbreeding. Gene flow, that means you have got these two population, these two population of same species, but the gene pool can be different. So maybe this one got a lot of E one, okay, a lot of I, no big A, maybe got small a, okay, no big A. So you got two populations here. Gene flow means that when individual from this population interbreed with individual from this population, so it causes a gene flow between these two populations. So gene flow means interbreeding. So therefore, when individual from here comes into this population, interbreed with individuals here, it may bring in a new allele from this population. Let's say, for example, no more E because of migration. Then after that, this E comes in and interbreed with small a. Then it produces small a, small e. Then becomes, there's a gene flow. And then the small e is introduced into here. So again, change in allele frequency. Mate selection also. Mate selection means when there's non-random mating, it will cause change in allele frequency. Why? Let's say, for example, you got big A, small e, small I in this population. But the A likes A only. Because E and I look very different, even though they can interbreed. So the A will choose A only. Big A choose small A choose big A. They don't choose E and I. This is called non-random mating. So you see that because of non-random mating, because there's a mate selection, then the allele frequency of big A and small a will increase, right? And then the small e and small I will decrease. See, this is called because of non-random mating. So all these processes here, not in syllabus, it's very easy to know that why they can lead to change in allele frequency. Okay, so today we're going to revise how selection lead to change in frequency, and then we look at genetic drift and founder effect. So selection recap, because we have learned natural selection, so we're going to recap that whole natural selection. So in the entire population represented by this entire table here, whenever selection pressure act on this population, it will then be able to divide the population into two big groups, right? The group on the left side are considered as the fittest organism. Fittest means they are able to survive. Okay, fittest in this case means when selection pressure act on them, they are able to survive because they contain favorable phenotypes. Favorable in the sense that they can survive in that selection pressure. So favorable phenotype, what is phenotype? Physical, physiological, biochemical, and behavioral characteristic of the organism. Okay. Hence, genotypes and alleles. So favorable phenotype, favorable genotype, favorable allele. So they are best adapted to the immediate environment. So therefore, they are selected for survival and they reproduce. So they have selective advantage. So when they reproduce, they pass on the favorable allele to the offsprings. So that will actually increase the favorable allele frequency in the population. And the favorable genotype frequency will increase. That will cause a change in the gene pool, which is all the different type of allele and genes in the population will change. So this is an entire explanation of the natural selection process. Okay, so the opposite will occur for the less fit organism. So you can see that how selection, which is in this case a natural selection, leads to change in the allele frequency in the population, where the favorable allele frequency will actually increase. Next, we're going to look at, so in a natural selection, selection pressure select fittest organism within population to survive and reproduce. Okay, that is natural selection. What is genetic drift? Definition. Drift means there's a change, a slight change, drift. So drift away from somewhere, right? So change in what? Allele frequency. And this occurs by chance in a small population. So this one occurs in a small population. From generation to generation, there's a slight change in the allele frequency. Due to what? A, founder effect, B, bottleneck effect. So these two will lead to genetic drift. That means there's a slight change in allele frequency in a small population. This one is for small population. Okay. So what happened? So this is the original population. So you can see there's variation here in terms of blue color and yellow color. So to show the variation. Then we know that some incidents happen. Some external factor happens. So what's the incident here? So it's a critical event that can cause a change. It could be some physical barrier that suddenly separate a group of a small group of individuals from the original population. For example, there's a flood, right? So this flood actually sweeps some of the individuals away from the original population. So it's being swept away. Or there's a flood and then this the original population is here, but there's some water that actually cut across and the flood is around here. So this group of individuals here are being separated away from the original population. Okay, so or forest fire also the same thing. If you got fire here that starts and cut across this whole forest, then it's separating between these two already. Okay, so natural catastrophe leading to physical barrier and causing the small group to wander away from original population. So they have to be separated from original population. They go to a new place. That's why it's called the founder effect. Founder, remember founder, this word is used to denote like when you discover a new land, right? And then you start to stay there. So you're the founder of that place. So this group of organisms, they are separated from the original population because of some natural catastrophe like flood, forest fire, and make them move away to somewhere. So therefore, it become a new population in another location already, separated from original population. Okay, it could be birds from the mainland, and because of hurricane being blown by the wind, and then they arrive to an island that's nearby. So they become a new population separated from the original population. So this is called founder effect. Okay, so what can we see here is this group of population here, their allele frequency, is it same as original population? Not the same, right? Because it depends on what are the individuals that have been separated. It's by random, so it's by chance. So even though this group is a sample from original population, but the allele frequency here will be different from original parental population, not parental, original population. Okay, so this is the meaning of founder effect. change in allele frequency that occur by chance in a small population. In this case here, because of some physical barrier, a small group wanders away. Right? Bottleneck effect. Bottleneck effect is slightly different. Bottleneck effect is, this is the original population here. And bottleneck means, when you say bottleneck, is like traffic jam, right? You come to a place where the road is very narrow, but there are a lot of cars, or 4 lanes become 1 lane, 4 lanes become 2 lanes. So that part is called bottleneck. So here what happened is that we always refer to survival. Something happens here that causes a lot of the organisms unable to survive. They die because of some selection pressure here. They die. So whatever left to survive, they will be a sample from original population, but the allele frequency will be different because it depends on who survived, right? It depends on who survived. So this group of new population here is a sample from the original one, but the allele frequencies are different from original one. So this is called the bottleneck effect. In both cases, there will be a genetic drift, means a slight change in the allele frequency starting from original small population. Because small population subjected to more change than bigger population. So this is the meaning of genetic drift and founder effect, and also bottleneck effect. Okay. So a small group colonize a new area and are separated from the rest of the larger group here. Population size is reduced by catastrophe. So this one, population size is very much reduced. Okay. Okay, what about this one here? Founder effect. So this is in elaboration of founder effect. Founder effect is the genetic differences between original population and isolated offshoot. So these 3 populations are small population. They have actually been separated from the original parental population because of some events. Okay, so they become isolated from original, so isolated offshoot. So there are genetic differences between this and this because it's by random who separate. So all of them you see are from original, but their allele frequencies are all different. Okay, so that illustrates founder effect. It focuses on change in allele frequency. Yeah. So alleles in the founder members of the new population being unrepresentative of the alleles in the original population as a whole, because this is only a sample. When you take a sample from original, it cannot be exactly the same. So it's slightly different. Okay. So therefore, the allele frequency of new smaller population is different from the original bigger population. As they are only a sample of the larger population, the allele frequency differs from the larger population. So when you take a sample, it is not exactly the same as original. So a slight change is happening here in the founder effect. Okay. So that's all for selection, genetic drift, and founder effect causing a change in allele frequency. So the next part of today's lesson is to do some mathematics. This is very easy. This mathematics is I think it's Form 1 mathematics. Remember you learned about p squared plus 2pq plus q squared equal to 1? Form 1, right? Hello? Still Form 1. I remember I learned Form 1. Hello, Form 1 mathematics. Okay, now we're going to do some Form 1 mathematics. Nice, right? Okay. So learning outcome says students should be able to use Hardy-Weinberg principle to calculate allele and genotype frequencies in the populations and state the conditions when this principle can be applied. So very easy. You want to find out what is allele frequency? What is the genotype frequency? Okay. So in order to apply Hardy-Weinberg equation to calculate the allele and genotype frequency, there's a rule to apply on it. The rule is can only be done on large population, because large population, the allele frequency more or less constant, right? Small population can change very fast. So you want to predict only large population, we can predict because it's quite random in the allele frequency. So in a large randomly breeding population, it means they freely mate with each other, not selective. Allele frequency remain the same from generation to generation, assuming that, so allele frequency remain the same because there is no selection, because natural selection, the environmental factors are the same. So there's no change, okay? No genetic drift, no founder effect, no mutation, no migration, no gene flow, no mate selection. That means no non-random mating. So when all these things do not occur, and when the trait that you are interested in is controlled by 2 alleles, then we can use Hardy-Weinberg equation to calculate the allele frequency and genotype frequency. So this is the condition for us to apply Hardy-Weinberg equation. Okay, large population, no events that can cause changes to them. Okay, so let's do the mathematics together. When a particular phenotype is controlled by two alleles of a single gene only, like our eye color, in this case, a big A, small a only, the population will be made up of three genotypes, right? Big A, small a, and small a. Calculations based on Hardy-Weinberg principle allow the proportions of each of these genotypes in a large randomly mating population to be calculated. So we can calculate the genotype frequency, the proportion of genotype. Okay, so the proportion of genotype is also the frequency of genotype. Proportion is frequency, is similar to percentage. So the frequency or the percentage of a genotype is its proportion of the total population. For example, big A, small a, and small a. Proportion of all of them add up is 100%, right? Okay, so this is called proportion. So the total is the whole population. So the total, when you add up the proportion of this, and this will be exactly 100%, which is equals to 1, represented by 1. Okay, and the frequencies are given in decimal place. This like probability, right? Probability 100% is 1, right? 25% is 0.25. Okay, so we use the letter P, small p, to represent the frequency of the dominant allele. Okay, big A in the population, and we use the letter small q to represent the frequency of the recessive allele, small a. So therefore, since there are only two alleles of this gene, so P plus Q must be equals to 1, isn't it? Proportion of dominant allele plus proportion of recessive allele added up must be equals to 1. So P plus Q equals to 1. Can I? Okay, next. So the chance of an offspring inheriting a dominant allele from both parents. So that means in a population, we have got small p to represent proportion of dominant allele, small q to represent proportion of recessive allele. So for an offspring, what is the chances of the offspring inheriting dominant allele from both parents? Is the chances of inheriting dominant allele is P, isn't it? From one parent in the population, the chance is P, and another parent is also dominant, is also chances is P again. So what is chances of offspring getting big A and big A, the dominant allele? So among the population, chances of big A is P, and cross with another chances of big A is P. So chances of offspring being big A is P squared. Okay, can everybody? Can. Okay, next. What is the chance of offspring inheriting a recessive allele from both parents? That means what is the chance of an offspring being small a and small a? So then you have to see, so what is chances of inheriting small a allele from the population, right? So chances of small a allele is Q, isn't it? Cross with, what is another chance of getting a small a allele from population? Q. So Q squared. Okay. So chances of an individual with small a in the population is Q squared. Next, so what is the chance of an offspring inheriting a dominant allele from father and a recessive allele from the mother? So again, what's chances of dominant allele in the population? P. Chances of recessive allele population? Q. So therefore, answer is PQ. So that means what is the proportion of dominant, heterozygous dominant, correct? Not heterozygous dominant is PQ. What's chances of homozygous dominant? P squared. What is chances of homozygous recessive? Q, Q squared. Okay. So based on our mathematics in secondary school, Form 1, right? Form 1, Form 2, Form 3. Form 3, you learn Form 3. Okay, Form 3, P square plus 2PQ plus Q square equals to 1, correct or not? Correct, This is mathematics, okay? So putting everything together, this is the equation. So the equation number 2. Equation number 1 just now is P plus Q equals to 1. Okay, so these two are important equations. So next, using these two equations, we can calculate the genotype frequency already of this population. Okay, so let's see. The homozygous recessive in a population can be recognized and counted. Let's say, for example, you got brown eye and blue eye in your class. Brown eye, I don't know, right? Because brown eye can be what? Brown eye can be big B, or big B, small b, right? So I don't know. But blue eye, straight away, I know it's small b, small b. So just look around and count all. What is the number of blue eye? And then convert to proportion for the whole population. Okay, then I know, okay, so in the whole population, right, I find that the incidence of small a is only 1 in 100 individuals. Like in your whole class, only 1 student got blue eye. Then I know what's the percentage of blue eye student? 1 out of 100. So it's 1%. 1% is 0.01. 1% is 0.01, correct? So therefore, small b. So what is the proportion of small a? Is represented by Q square, isn't it? Q square. So Q square is actually the frequency of homozygous recessive in this population. We have discovered in the population is 1%. So 1% is 0.01. So Q square is equals to 0.01. Therefore, Q equals to 0.1. Okay. So already found out what is Q already. Q is 0.1. So Q is the frequency of the recessive allele in this population. We found already, right? Q is frequency of recessive allele 0.1. So we found already. So then, can we find P or not? Yes, we can. Because P plus Q equals to 1. Correct? So I found Q already, I can find P. Find P. P is equals to 1 minus Q. So it's 0.9. Correct? P is 0.9. So I found these two already. This important information. Q is 0.1, P is 0.9. So P plus Q equals to 1, right? Okay. P square, you square is 0.81. So how many percent of individual here is homozygous big A? Homozygous big A is P, isn't it? It's 81%, correct? Correct? So therefore, big A, percentage of big A is 0.81%. Because frequency of big A is 0.9. Frequency of big A is 0.9. So 0.9 times 0.9 is 0.81. Okay. So what about 2PQ? So they want to know what is 2PQ. 2PQ is the proportion of heterozygous. Okay, because we already know from equation that P square plus 2PQ plus Q square equals to 1, correct? So that means to say, if P square represents the frequency of homozygous dominant and Q square represents the frequency of homozygous recessive, then 2PQ must represent frequency of heterozygous, because when all the proportion add up, you give you 100% or 1. So what is the frequency of heterozygous? Is 2PQ. So therefore, 2PQ is how much? Why the calculation like that? 2PQ means 2 times 0.1 times 0.81, right? Correct? No. What is it? They calculate in two ways. You see, they calculate in two ways. You calculate yourself. Is 2PQ equals to 0.18? Calculate. Is 2PQ here equals to 0.18 not? Yes, right? So it's correct. It tallies with this equation, right? It's actually correct. Okay, it's correct. Or there's another way to calculate is that I know Q square equals to 0.1, right? And I know P square is equal to 0.81, right? So whatever left must be heterozygous, isn't it? So therefore, they take 1 minus 0.1 minus 0.81 will still give you 0.18. Two ways of calculating. Okay, so there are two ways to calculate. So both ways are correct. Okay. So now, are we ready to do question? Yes. So recap. The Hardy-Weinberg principle. So in Hardy-Weinberg principle, he said that the allele frequency of a dominant allele in a population is represented by P. And the allele frequency of a recessive allele, I think you need to fill in something in your notes. You go and check your PowerPoint. So the allele frequency of small a is, the allele frequency of recessive allele is represented by Q. So P plus Q equals to 1. This write into your notes. P plus Q in your PowerPoint. P plus Q equals to 1. Okay. Then for genotype frequency, frequency of big A is P square. Frequency of big A, small a is 2PQ. Frequency of small a is Q square. And P square plus 2PQ plus Q square equals to 1. Correct? Okay. Okay. So phenotype frequency. So now if you know genotype, we know phenotype, right? Okay. Phenotype frequency. What is the frequency of people showing dominant phenotype? Is the big A and big A small a, right? So therefore it's P square plus 2PQ. So once you know the genotype, you will know the phenotype frequency. Recessive, Q square only. Okay. Okay, so we try out question. So example, let's do this together. 4% of the population suffers from cystic fibrosis. Recessive, right? So cystic fibrosis is coded by a recessive allele of the CFTR gene. So that means to say the Q square, isn't it? Q square, right? Is equals to 0.04. Correct or not? Where's my 0.04? 0.04. Correct? Then you do the calculation. What they ask you to do? Okay. So question asks a student, what percentage individuals have the normal phenotype? So how many percentage of the individuals have normal phenotype? Can you calculate now? Of course, very easy, right? The rest have no cystic fibrosis, right? Which is 96%, so it's 0.96. So this one, very straightforward. Okay, so write down your answer, please, into your PowerPoint notes. Next question. What is the frequency of the dominant and recessive alleles in this population? So can do or not? Q square is equals to root square, right? Root square of 0.04 is equal to 0.2. Right? P squared equals to, P plus Q equals to 1. So P is equal to 1 minus 0.2 equals to 0.8, correct? 0.8. Okay, so answer is 0.8. Okay. Yes, answer is 0.8. Say C. What percentage of population is heterozygous for the two trait? Oh, very easy. 2PQ, isn't it? Correct? It's equals to 2 times P times Q. So what's the answer? How many percent? 0.32, so 32%. Sounds very straightforward. Easy, yeah? Can do, huh? Okay, so let's try the next question. This one is interesting. It is thought that the coat color of a particular type of rodent varies between tan, which is dominant, brown is recessive. Assume the population is in Hardy-Weinberg equilibrium. You observe 336 tan rodents and 64 brown rodents during your study. So that means these 10 rodents, right? You have got this is coded by dominant allele, right? So it's homozygous dominant and heterozygous dominant. Recessive one is small t, small t. Okay, so A. What is the frequency of the homozygous recessive genotype? What is the frequency of homozygous recessive genotype? Homozygous recessive is 0. Sorry, it's not 0.64. It's 64, right? Altogether got 64 divided by, hello, didn't come up. 64 divided by total population size 336 plus 64. Correct? So that's the calculation. Answer we check later. We see calculation first. Okay, can. B. What is the allele frequency of the dominant allele in the population? Dominant, you have to use a recessive one to find out, isn't it? Okay, so Q square is equals to Q square is equal to how much? 64 divided by 336 plus 64, right? So therefore Q is equals to root square of, right? Root square, right? Of 64 divided by 336 plus 64. So therefore, P is equals to 1 minus Q. Correct? So that's the calculation. So what is the actual answer? We check later on. Okay? Can everyone? Answer we check later on. C. Of the animals you observe, how many were heterozygous? Very easy, right? 2PQ lah. Correct? How many were heterozygous? 2PQ. So I already got P, I got Q. I can calculate 2PQ. Okay. Answer we check afterwards. D. How many of the 650 animals? Do we have 650? Oh, okay. This is a different generation already. Okay, this is different generation. How many of the 650 animals would you expect to be 10, assuming the population is still in Hardy-Weinberg equilibrium? So this is another generation already, because the total number is 650. The original number is actually 400. Okay, so in another generation where population size is 650, how many of them you expect to be 10? That means 10 means how many TT and TT, right? Okay, so that means P square plus 2PQ is the answer, isn't it? Okay, Can everybody? After we see the answer. Last question. As you observe the animals, you count. Wait, my P square and 2PQ for this one is different, isn't it? Hey, hello? My total number is 650, you know. Actually can. P square plus 2PQ. How can P square plus 2PQ? I must do something to this, right? Hello? I must do something to this, right? Convert 400 to 650. No. Then do what? I forgot. And because your original, the proportion is correct, but now you're referring to 650 already. So you have to times 650 lah. Correct? Times 650, yeah? So this one, I think you have to times 650. Okay. Okay, last question. As you observe the animals, you count 200 brown, 450 tan. Conduct a chi-square test to determine if your observations are significantly different from what you expected. Wow, very good. Eh? So can you tell me what is your expected ratio? Expected ratio for brown versus tan. I don't like brown and tan. I like tan and brown. Because tan is usually the dominant I put in front to brown. So what is the expected ratio for tan to brown? Your tan is P square plus 2PQ, right? Okay. Times proportion means how many in total? Hey, sorry, what am I doing? This is 10, correct? This is 10 times 450 plus 200. Your population size is this big, correct? How many of the total population size here have got 10? Is this one, right? Because this entire population size, 450 here plus 200 is the total. So this is the percentage of the total of this having 10 color, correct? To brown. Brown is Q square times total. Okay, can? This part you can or not? Hello? Because your total number of your expected must be the same as your observed. So to make it the same. Observed 10 is 450 to brown is 200. So your expected ratio, right? You add up all must be equals to 450 plus 200, same as this one. Then only you can compare between the two. So you make the population size of the E and the O must be exactly the same to compare. Okay? All right, shall we check answer? We check answer, huh? Okay, so this is the answer. Please check. You can pause your screen to view, because I'm going to pause. Okay, so now we already done a Hardy-Weinberg calculations, but we need to know that these Hardy-Weinberg calculations, they do not apply when the population is very small. Remember just now there's a condition where it can be used. So when is it cannot be used? When the population is small, or when there is a selective pressure against one of the genotypes. So there's a selection happening. We cannot use it. Migration also we cannot use it. Non-random mating cannot use. Okay, everything that can cause a change in allele frequency, we cannot use it. Because once there's a change in allele frequency, our calculation is no longer correct because we are using assumption one. Okay, assumption that the allele frequency remains stable, remain the same throughout generations. Okay, nothing should change them. Then we can apply this in large population. So what is the use of this calculation? When the ratios of different genotypes in the population have been determined, their predicted ratios in the next generation can be compared with the observed values. That means in this generation, we already calculated the percentage. Then in the subsequent generation, we can also go to the population and calculate percentage and compare. Is there a difference between this generation and the next generation and the next generation? Using chi-square, we can compare, right? Okay, so after compare, we can see whether there's any significant differences when we do the chi-square test, comparing this generation and next generation. Is there any significant differences? So if there's any significant differences, if the differences are significant, okay, and we know that there's no migration, no non-random mating, then there could be due to some directional selection. That means frequency of change, right? What causes the frequency to change really? Okay, if there's a significant difference between observed and expected, observed in the current generation, expected from previous generation, we calculated, there's a difference. And then there's no migration happening, there's no everybody is mating randomly. Then something else happening. It could be directional selection. Because if it's founder effect and bottleneck effect, we know one, right? Because natural catastrophe, a lot die away, but now population size remain the same. Because founder effect and bottleneck effect, the population size becomes very small one, then we know. But this one, population size the same. But how come change? Natural selection, directional selection. Okay, so there's a change. So this is a conclusion we can make after we do the chi-square test. So recap, what is the assumption of the Hardy-Weinberg equation when we do the calculation is organisms are diploid. Yeah, that means you got a pair of allele that code for the phenotype, it's diploid, and they're sexually producing. Generations are non-overlap. So when you take reading from this generation, you take reading again, maybe like 10 years later, we take it as a next generation already. Okay, assumption, this assumption. Usually it doesn't happen, right? You say generation doesn't overlap, that means your father, your parents, and you we don't calculate together or no? Cannot. Okay, so this one is just assumption. Mating is random. Population size is infinitely very large. Allele frequencies are equal in the sexes. No sex-linked alleles. So allele frequencies are equal in both male and female because it's not sex-linked. That means this allele must not be sex-linked one. Okay, it's in autosome, not sex chromosome. There's no selection, no mutation, no migration. So that's the assumption if you want to use Hardy-Weinberg principle. So this is a past year question. This is called what? Resistance to, what is this? Resistant to malathion would be dominant to the non-resistant. So resistant to this malathion, which is a chemical, is coded by dominant allele. Okay, whichever that's resistant to malathion is homozygous dominant or heterozygous dominant. Okay, so what does the question ask the student? Use the Hardy-Weinberg principle to calculate the frequency P. That means the dominant allele, right? Of the allele for resistant in Jamnagar in 2005. So look for the data 2005 is here. Percentage of mosquitoes, so talking about mosquitoes, mosquitoes being killed by malathion is 76%. So are we able to find out what is P? Can, Can, right? Hello, please calculate and tell me your answer. P square plus 2PQ. 2PQ is equals to 76%, right? 0.76, correct? Oh, no. Cannot. I cannot use that one. I must use 24. Q square is equals to 24. 0.24, correct? I must start from here, right? So therefore, P is equals to 1 minus Q. That's how you find out, correct? So what's the answer? Yay. Correct. Is the answer correct? Is the calculation correct? Oh, okay. I have to leave already. So that's all lah.